So our last speaker today is Dr. Bart Weimer. He's a professor in the Department of Population Health and Reproduction at UC Davis at the School of Veterinary Medicine. He's also director of the 100,000K Pathogen Genome Project and BGI at Davis. Dr. Weimer's group is focused on microbial physiology and function, and they employ large-scale genomics and systems biology approaches to understand the complex interactions between humans, food, animals, and the environment. Specific interest areas include antibiotic resistance, the gut microbiome, and how gut bacteria interact with the immune system via stem cells. Dr. Weimer has over 24 years' experience in transforming advanced genetic information into actionable knowledge. He's authored more than 110 peer-reviewed publications and book chapters, authored 3 books, and has mentored over 30 students, many of which I've had the pleasure of working with over the past few years. So I'm going to hand off to Dr. Weimer, I guess.
First of all, thanks very much for the invitation to be here today. This is not a meeting I typically go to because I focus primarily on bacteria sequencing opposed to higher animal sequencing. So my talk is actually going to be split into sort of 2 topics today. One is about microbial sequencing and high throughput sequencing, and I actually got my start with JGI sequencing genomes in 1999 was the first project. The CSP program had just barely started, and we were the first packaged genome project that got funded, and it was 12 genomes, and it took us 3 years to sequence 12 genomes. Today, we're not doing it quite that slowly. So my group is pretty diverse. So when people run into me, sometimes I'll be talking about microbes, sometimes I'll be talking about systems biology, sometimes I'll be talking about metabolism. And so today I'll tell you a little bit about the whole genome sequencing we're doing for microbes, and then I want to tell you about a story that we're quite excited about, and it was enabled by a couple of, well, an extensive collaboration with Kappa, but also some other companies as well. And so this little diagram sort of shows how we view the world, and so each of the little red boxes we study is something in that area. So I want to start with Salmonella, and primarily because the 100K project, which I'll describe more thoroughly, is really focused, and it was focused initially on foodborne pathogens, because foodborne disease in this country and across the world is not going down, it's actually going up. And so that's quite a surprise for many of the folks around the world. And Salmonella in this country, we have about 4 to 6 outbreaks per week of Salmonella in the US alone from a variety of different products. So if you actually set up a Google alert on Salmonella and outbreak, it'll pop up 5 to 6 outbreaks almost per day on your Google account. So we got into this as a large-scale university, industry, government collaboration. And recently, the government has now started to use next-gen sequencing in regulating foodborne outbreaks. So they've stopped collaborating essentially with academia, and now they're using it in a regulatory role. We're still progressing on. So we got into this because in microbial sequencing, most everybody uses 16S genes. It's the common gene to name a microbe. When you do 16S or pyrosequencing with this gene, there's a lot of SNPs across the 16S gene across a lot of organisms. And this is just a handful of Salmonella that we picked up, and we just did an alignment, a tree of 16S. And while it's a slowly evolving gene, there are lots of SNPs that accumulate. Salmonella is a modestly quickly evolving organism. And so that wasn't really informative. So as we started to sequence Salmonella, and others are sequencing Salmonella, then we started to be able to have the ability to do whole genome alignments, and then all of a sudden the tree started to make sense. Because Salmonella is named on a serotype level, so that's almost like a twin in a sense. From a human perspective, a serotype, the genetic similarity of serotypes are very close. You would think. And in fact, they're actually not that close. The genotype doesn't correlate to the serotype. But in epidemiology, the serotype is so widely used to name the organism, and serotyping is controlled by the World Health Organization. So it's not going away anytime soon. And PHYLOGENOMICS is probably going to start pushing on the door to replace serotyping. But it was very quickly understood that these red boxes, they should be aligning, or they should be coming into the same clade. And when you look at 16S, they're not, and when you look at whole genomes, they are. So that signals to us straight away that we should be using a lot more genomics in this area. So when we started doing this, well, in today's world, there's about 2,600 serotypes of Salmonella alone. E. coli, there's about 500 or 600. And then other pathogens, there's a variety of levels of serotypes that are referred to. There's an extensive amount of horizontal gene transfer going on in microbes that you wouldn't expect to occur. Increased stress increases horizontal gene transfer, and I'll show you that in a couple of studies in just a moment. And then we were part of some work where all of a sudden, not all of a sudden, it's been happening, but we were able to define hypervirulent salmonella are starting to emerge. So they go from an animal to a human and back to the environment or back to the animal, and they become hypervirulent. And we've just finished a bunch of sequencing of 16 different genomes of these hypervirulent organisms to pinpoint exactly which loci are causing the hypervirulence. And ironically, it's related to metabolism and stress response in most cases. So also the piece that's coming to play, especially in the food area, especially on plants and the microbiome of plants, is abiotic stress regulates the plant, but it also regulates the microbiome and who's there and what they're expressing and the interaction between the microbiome and the plant. And we've recently published a couple of papers looking at temperature and acid stress and the increased persistence of Salmonella both in vitro and in vivo. So there's a group, Eric Alm's group, who used to be out here on the West Coast and is now at MIT, did a nice study with Vibrio. And Vibrio is a very diverse organism that lives in the ocean, tends to be accumulated in seafood, especially in oysters. And so as you know, oysters are filter feeders, and as they filter the water through to gain the nutrients, they also accumulate Vibrio. And the oyster beds in the North Pacific Northwest and in the Gulf of Mexico, which are just about gone now, but a small shift in the ocean temperature shifts the type of Vibrio that associates with the oysters. And so they did this beautiful study showing that if you start sequencing these Vibrio, there's footprints of biogeography within the genome of Vibrio. So where it grew up, so to speak, you can start associating different loci with where it originated from. They were also made claims of co-evolving gene sets so that you could identify who their neighbors were. So that has implications for metagenomic projects, especially related to soil and ocean. So the bottom line of this, which is actually no big surprise, is that stress increases SNPs, and bacteria accumulate SNPs if they've got mutations in a replication loci called mute. And they can happen at a very high rate. We sequenced a gram-positive organism that had a mutation in one of the 3 genes that are responsible for repair in that organism, and it had the highest mutation, had the highest SNP count that was seen at that point. And so these horizontal gene transfer events occur commonly, and the recombination events occur commonly. So as we continue to stress organisms, and in the food industry, the very first thing we do is throw acid on something or we heat it up. And those are exactly the 2 things that induce stress response in the microbe, induce SNPs, and induce persistence. So some of you are familiar with the outbreak in 2010 of E. coli in Europe, which was represented by a chimeric E. coli genome. So there were 2 pathotypes that hadn't been seen since the 40s. They formed a chimera and re-emerged, and so there was a lot of trouble solving that outbreak. So we started the 100K Pathogen Genome Project in 2012, and it really was the goal to, if we're going to have next-gen sequencing be part of regulation or by the government, then we better have a good reference database or a reference set of genomes that are out there. And in fact, since 2012, there were, between all of the enteric organisms, there were about 2,500 genomes available, and today there's about 15,000 genomes available. And so these are the sorts of organisms that are being sequenced in the 100K. There is an active FDA program that's going on across the entire U.S. where they've got 10 sites that have MySeqs, and they're producing sequences every day, or every month, I should say. And they do about 500 sequences a month now, and they upload directly off the back of the MySeq onto NCBI. So every year, you're going to start seeing Salmonella, E. coli, Listeria are the top 3 you're going to start seeing 15 to 20,000 new genomes per year based on FDA and the 100K combined. And then CDC is now starting to do this as well, so it depends how many CDC starts to sequence. So the take-home note is that it's distributed sequencing at its infancy, I suppose, right now. The EU is also implementing next-gen sequencing in this area, and now it's not a question of who's going to do it, it's only a question of when it will be implemented in that particular country within the EU. So we've focused most of our effort on Salmonella because that's the biggest problem in the US, but last year we sequenced just over 8,000 of these organisms and approximately 1,000 of these organisms, and we're in the midst of either giving them back to the originator, the sequences, or we're posting them to NCBI. Okay, so our pipeline essentially is we mandated isolates be sent to us. My experience with JGI was you isolate DNA and it's not good enough, and you isolate DNA and it's not good enough, and their standards for quality are very high and it's very good. I didn't want that to happen in this project, so we had them send us the isolate, and then we isolated the DNA and did the downstream processing and gave the sequence back from them, to them. So now we're really at this place where distributed functional genomics can start occurring. The point of this is to create reference genomes for broad-scale use, and then also to enable novel discoveries. I was just in the UK at a meeting about antimicrobial resistance, and the entire world is starting to be worried about this. And the UK Minister of Health, she predicted that, or they had just released a report predicting by 2050 that death from multi-drug resistant bacteria would be the leading cause of death across the world. It would outstrip cancer by far. So we're running out of antibiotics and we're quite worried about it. Okay, so this is our protocol that we work through. We authenticate the cultures. We had about 20% of them, 15% of them contaminated. We run through, we use the Kapa's traditional system, I'll call it, traditional library system, where we used sheared, Covaris sheared DNA going into it. And then we have the ability to make about, between the robots, we have 300 to 600 libraries per day in a 96-well format. Most of the sequences are going on Illumina, and a few of them are going on to PacBio. And then we're just in the midst now of, it's actually formally announced that we've expanded the 100K to China, and it's a collaboration between Davis, Beijing Normal University, City of Beijing, and the Academy of Science, Chinese Academy of Science of Microbiology, where they will take on sequencing 10% of our goal. So they'll sequence 10,000 genomes in the coming year. And that's already up and running, and their goal is to look at traceability, diarrheal diseases, and antibiotic resistance. So you can imagine if you're at JGI, you absolutely need to have a standardized protocol, and so do we if we're going to have a produce reference genomes. So we identified areas within the workflow that we absolutely needed to standardize and streamline, both for cost and for throughput and for timing of people. So most of what we're doing is becoming automated. So if you just look at the shearing step, we had already run into a problem of shearing for this project, because if we're doing 10,000 genomes per year, I didn't buy my own Covaris, and so now I have this problem of where do I find it? I run to my genome center and they graciously run my samples for me, but the costs are fairly high and the throughput's fairly low. So to sequence to shear a half a plate for sequencing library takes me 8 hours, so it takes me 2 or 3 days to shear enough DNA to make enough libraries. So clearly too slow. So in the midst of that, we were collaborating with Agilent at the same time, and we produced a whole series of application notes to sort of lay the land of put the stake in the ground of what we're doing and how we're standardizing our methodology. So we developed an app note to define a DNA extraction, because the microbes that we were working with are so diverse, and we needed to have a single step that would lyse everything. So we developed that technique. We also used one of their newer instruments, the tape station, to do whole genome or genomic DNA validation, which actually this app notes are out and available. And then we worked with Kapa to produce a QC protocol on the tape station, and then we also worked with Kapa and Agilent to produce a automated library construction protocol, which is within this app node as well. And so that actually gives us the stake in the ground of what we're doing, and we haven't varied from these app notes. We're still doing the same thing. So you did a great job previously of showing what, the coverage on the sequencing. We also made these are just some tracings of whole genome libraries with the new HyperKit with enzymatic fragmentation. And these are 100 nanogram inputs made with automation as well. And so each of the lines is a replicate, and it's looking pretty good. The GC content is the big deal here. And so the challenge for us is to absolutely make sure that the GC content doesn't skew the library or doesn't have caused problems in library construction, which it didn't. Within the sequencing area, everything looked good. We got great consistent coverage across the genome for no GC bias that we found. And that was a big deal for us because a large proportion of the genomes that we're sequencing today are about 32 to 36% GC, and some of the other organisms are 65 and 70% GC. So we wanted to have a common platform that we wouldn't have to vary based on GC content. Okay, so let me launch into a couple of vignettes of what we've done with the sequence once we have it. So the whole background here is that next-gen sequencing is moving quickly into the food safety and food security space, and especially food safety. And metagenomics is coming along very quickly. And so because of the time constraints, I'm not going to show you any of the metagenomics work that we're doing with the same library construction kits that we've already been talking about. So CAMPYLOBACTER is an organism that's very small. It's related to Helicobacter. It's extraordinarily mutable. The genome is only about 2.1 meg. It's very tiny. So this project is scaling up now to sequence 386 or 384 per lane on a HiSeq. We've been indexing already at 96 per lane on the HiSeq, and we're getting really good coverage across 5 megabase genomes. So we now have everything in hand to do this in a much larger scale, and so we need to be able to make libraries at a much faster pace. So what I wanted to show you today is that we were able to identify some extensive genome rearrangements within this very small genome. While this genome is very small, it's littered with mobile elements. It's about 25 to 30% of the genome is mobile elements, and so it can lead to some extensive rearrangements. And so those rearrangements then allow us to segregate them into the species. And this is some sequencing that we just recently finished to show that jejuni is the most common human pathogen, but it's also been shown to be a diarrheal agent in non-human primates. And so we were able to obtain some samples from the primate center that we have at Davis. It's also a very common commensal in crows. And if you've ever been to Davis, Davis is full of crows. And in fact, in San Diego, I've noticed there's quite a few crows. And CAMPYLOBACTER is a non-pathogenic commensal in most birds, in poultry as well. Coli is a pathogenic group, and Leri is a unique group that tends to be only in primates. So let me now switch to another organism called Listeria, which is a foodborne pathogen as well. It's a gram-positive. The genome's about 2.5 to 3 meg. It's known to be fairly stable genomically. We were able to sequence a few of these organisms and show that they had some inversions going on in the genome. In almost every case, these inversions were being manipulated or being facilitated by tRNA locations. So we found no other mutations within the genome other than the tRNA. So I gave you two examples of extremes of CAMPYLOBACTER where it's extraordinarily mutable. We were able to find that. And then Listeria where it's not very mutable. And we were able to find that and identify exactly where they are in the ribosomal subunits. Okay, so what we were able to do with the initial work we were doing with Kappa is sequence about 9,500 genomes with shearing, and then automatic library construction. We're now moving to the enzymatic kit, the HyperKit, to finish off in second year. We've planned locally we're going to sequence about 6,000 genomes at Davis this year. And in China, they'll sequence 10,000 genomes, and we're in discussions with a couple of other groups. So we need to have a much faster methodology. So On the automated method that we're using today, we can make a nice enzymatic shared library in about 2.5 hours, and the QC is done in the next 30 to 45 minutes. So within a half a day, we've got a plate full of libraries ready to send off to the sequencer. Okay, all right. So now let me switch gears and tell you about some work that I'm quite excited about, and there's only one other group in the world that I have actually seen trying to do this. So we were collaborating with a lady in the vet school who was interested in regenerative medicine and also gut inflammation and IBD and Crohn's disease. And underneath in your gut, the gut lining is only at one epithelial cell thin, or thick, if you will, and it's mostly mucin or covered by sugars. And so underneath the epithelium essentially are stem cells. And so as the cells slough off, stem cells generate and cover the make new epithelial cells. So a student in my lab who's just finished and is actually staying on for a little while, who's headed to med school in August, unfortunately. I'd love to keep her around. We started working to say, well, hey, what are the gut bacteria doing, and can they infect stem cells? And if so, what do they do to stem cells if they can associate with them? So we started off with this study to say stem cells do many things, and I won't go into all of the things that they do, but essentially they can mature into lots of cell types. They home to where there's inflammation, so they're very responsive to inflammatory cytokines and so on, and they produce their own, and they're pre or pro-inflammatory. So we started to put bacteria together, and our first publication came out at the end of 2014, where it showed both probiotic bacteria, normal flora in the gut, and pathogens actually associate with stem cells, and they invade stem cells. And we said, well, if they invade stem cells, I wonder what they do to them, because if you invade an epithelial cell, within about 4 hours, the cell's is induced to apoptosis, and it's lysing. Well, the short of the story is when Salmonella sees a stem cell and invades a stem cell, it alters the differentiation trajectory for that particular stem cell. So we've done it in different species. This is goat, and this is human mesenchymal stem cell invasion. So as the bar below the line represents invasion, the bar above the line represents association or binding to the surface of the stem cell. So you've got two localizations of the bacteria. Some are still on the outside, and some have actually got on the inside of the cell. So these particular plots are of different bacteria, actually different Salmonella. So it's not only species specific, it's the type of Salmonella that is associated. So it's incredibly variable. And there's some micrographs of what it looks like. And these are Salmonella on the inside of the cell, and every pathologist that I've talked to, and they look at this, they say they can't tell me that it's inside a lysosome or inside a vesicle like you would normally see Salmonella in an epithelial cell. So we're presuming that they're free in the cytosol of the cell. And in fact, if you want to see some other pictures, I've got some stem cells that are jam-packed. You can't see any of the cytosol because they're so full of Salmonella. So we found this very interesting picture where the cell looked as though it had actually produced something that the microbe is sitting on. So this is a piece of the cell membrane, and Salmonella is sitting down onto the surface of the cell. So it ruffles the cell, but it doesn't change the morphology too much. And it does significantly differentiate inflammatory cytokines within the stem cell. So here's a comparison of the apoptotic pathway. The important part is not what the steps are. The important part is how much red you see on the left side of the screen. And this is what happens in an epithelial cell. So Salmonella induces apoptosis by a variety of mechanisms within an epithelial cell. In a stem cell, there's virtually nothing. There's no induction of the apoptotic pathway by RNA-seq. So this is a whole population RNA-seq experiment. Same thing with TRLs, or toll-like receptors. It's very well known that bacteria bind toll-like receptors, and when you have epithelial cells, there's a whole bunch of regulation of the molecules below the toll-like receptors, and you see all sorts of toll responses, including inflammation. No such response has been found that we found in these are human mesenchymal stem cells. So whatever way they're getting into the cell, it's probably not through the TLRs. but they are clearly getting in from some way. So then we did this population, RNA-seq project and started to understand what sort of pathways or molecular pathways are being regulated. And so these down here are repressed and these up here are induced. And so when you start looking at the functions from the host side, edema, maturation of dendritic cells, and then apoptosis is actually repressed. So it looks as though bacteria using RNA-seq are actually keeping the stem cells alive or pushing them away from popping, I guess is the way to say it. And then just here's NFKB, which is one of the inflammatory pathways, and so you can see that there are things occurring, and they are regulating pro-inflammatory pathways across the entire stem cell. So we had this opportunity recently Fluidigm was on campus, and we thought, what we ought to do is compare what we see in single cell and what we see in population. So we took our infected stem cells with bacteria. We did flow cytometry into 3 separate populations, not low infected, medium infected, and high infected. So on flow cytometry, we segregated those, partitioned them and ran them on the C1 platform, and then put them into the HyperKit for library construction. Thank you very much, Kapa, for helping us, because we were very quickly moving along, and they were very helpful in getting us reagents. And we sequenced the transcripts, and then went off and used IPA to look at them. So here's an example of the flow cytometry, and that's what it looks like. It looked very good. Our libraries came out in 2 separate populations, which we were quite happy with. We could then pick which one we wanted to sequence. We looked at the QC, and again, we were quite happy. There might be a little bit of overrepresentation on one end, but nothing serious. We were quite happy. So everything looks good on the QC from sequencing. And we looked at the functional analysis, and EIF2 is actually a master regulator of inflammatory and a whole bunch of other responses within the cell. Rho is an inflammatory response as well. And so we're quite happy to see that we got lots of things related to inflammation and IL-8 signaling, thrombin signaling, and then here's a similar picture for NFKB. So they now have an entirely different NFKB network that's being regulated in single cells as opposed to the whole population. So we were quite happy to see this. We actually found a whole bunch of new transcripts that we couldn't see in an entire population study. And so using a single cell analysis network really actually helped us. So we were extraordinarily happy with the new enzymatic kit, the HyperKit. Faster, less expensive for us, and created data that we could actually find new transcripts. And then we're just in the very beginnings of understanding what microbes are doing to stem cells, and we're off chasing that very hard. And so I want to end there and thank my lab and my collaborators and Kapa for all of their help, and a shameless plug for a book I just co-edited if you're interested in metabolomics. Okay, thank you very much.